Hey guys. So in this lesson, we're looking specifically at cell signaling. So we're kind of doing an overview, and then we have another one. We'll go into some more detail on things. So when we're talking about cell-to-cell messages, we have three key stages. We've talked about the different types of messages, but now we're moving into three key stages. So the first stage is what we call reception. So the ligand, or the little chemical messenger, is going to bind to a receptor. Then we have transduction, where notice we got into the messenger, and so now we're going to convert that signal. And then response, the cell process is actually altered. So it causes the change in the new cell, or in the cell, that was desired. So looking more specifically, when we're talking about reception, so this is where we're detecting, we're receiving the ligand, that chemical messenger, by a receptor in the target cell. The target cell can be the same cell it originated in, or it could be a different cell close by, connected, far away. Remember, we had different options. So the receptor is typically going to be some sort of macromolecule that's going to bind to that signal molecule, that ligand, that chemical messenger. All receptors have an area that interacts with the ligand and an area that transmits a signal to another protein. So remember, that area that interacts, it's typically a lot like a puzzle piece, because we're trying to make the connection between that specific ligand and that specific receptor. And that typically will cause some sort of shift or change in the receptor to create that transmission point. The binding between the ligand and receptor, it's going to be really, really specific. We don't want it to happen to just anything. So when that ligand, and notice here, we've got this orange substance, this chemical messenger that was released from one cell, and it is attaching to the receptor, it activates through some sort of conformational change. So it's going to change the shape of that receptor somehow. This allows the receptor to interact with other cellular molecules. And so that's what that change in shape, that conformational change, is what initiates transduction signal. So that's what's going to cause the movement. Receptors can be in the plasma membrane, as shown in this illustration on this page, or they can be within the cell, because remember, some messengers, especially those lipid-based ones, can actually move directly into the cell, so they could be found inside of the cell. So what's the difference between the two? So in plasma membrane receptors, it is the most common type of receptor involved in the signal pathways. And it binds to ligands that are going to be polar, water soluble. They could be large. So things like G protein coupled receptors, ligand gated ion channels, these things are going to be controlled by that ligand. Intracellular receptors are found in the cytoplasm. So these are the ones that, or the nucleus, these are the ones that it can actually pass into the cell before we find the receptor. So it's going to bind to ligands that can pass through the plasma membrane. So things that are hydrophobic molecules, a lot of lipid-based, like steroids or thyroid hormone. Additionally, we can have certain gases that easily pass through, like nitric oxide plays a big role in a lot of different behaviors. So this is looking here at this picture with the intracellular. So notice that the signaling molecule, this little red circle, is actually able to move into the cytoplasm, where it then attaches to that receptor. And then once they are attached, they move into the nucleus to the DNA, where they will lead to their change. So the second stage is transduction. And so this is the conversion of that extracellular signal, that chemical from the outside, to a signal within. And so that's what we've got going on, showing the green on this diagram, to bring about some sort of cellular response. Because remember, the whole point of this is we want the cell to do something. Typically, when you have this, there's going to be a series of changes. And we call that a signal transduction pathway, and we'll talk about that in a little more detail in a different lesson. Okay. So that pathway is actually going to regulate all the protein activity as it goes. It may do it by phosphorylation using the enzyme protein kinase, so it can relay the signal inside of the cell, or it may do dephosphorylation by the enzyme protein phosphatase, two different enzymes for two different behaviors. And that one will actually typically shut off a pathway. Sometimes the signal is, hey, we don't need any more of this. Let's turn off this process. So that's like an on switch or an off switch kind of concept. Okay, so remember, the change in the shape means change in function. We learned that when we talked about proteins way back when anything that changes the shape changes the function. So during transduction, that single little signal that came in, we can actually amplify that signal. We use secondary messengers because notice our messenger's on the outside here. So it's going to come in and it's going to pass to pass to pass. It's like a big game of telephone to a certain extent. So one of the big secondary messengers involved in this amplification process is cyclic AMP, often referred to as CAMP. It is one of the most common secondary messengers that we come across when we look at some of these different transduction signals. And then the third stage is the response. So this is the final molecule in the pathway that converts the signal to a response. It's going to alter some sort of cellular process. Typically, we find it's either going to turn on some sort of process or it's going to turn off some sort of process, because most of the things that happen within a cell are very much just different chemical reactions that have been triggered. So it could be that the protein can alter the permeability of the membrane, allowing certain molecules to come in or not to come in. We talked about how a cell can actually signal and increase the creation of the bioluminescent material when we watch that TED Talk. There could be enzymes that are going to change a metabolic process. Or we could have proteins that turn genes on or off so that we are transcribing and making new proteins, or we're stopping making new proteins. Okay, so what are the three stages of cell signaling? Reception, transduction, and response. What is the actual signal being transduced in a signal transduction pathway? And that would be the ligand, that original chemical messenger. How is this signal passed from outside to inside the cell? And that is through transduction, that secondary system. During transduction, the signal is relayed by protein kinases and amplified by secondary messengers. So that's almost like that game of telephone. Okay. So hopefully that gives you a little extra help. I'll see you in the next lesson.